Okay, this is your video lesson over mitosis. You need to make sure you are writing these notes down in your journal. Make sure you're writing down anything extra that I write down on the screen as well. And that includes any drawings that I sketch on the screen or that I point to and tell you to make sure that you write down. So mitosis is the process of dividing the nucleus. Okay, so this is how our cells go through and sort the DNA and divide the nucleus prior to dividing the entire cell. This is a very, very important process that happens in all of our cells, with the exception of only a couple. And this is a process you're going to need to know backwards and forwards. So here we go. Mitosis is broken down into 4 phases or 4 steps, and step 1 of mitosis is called prophase. Okay, so in prophase, this is our first step, or you can say P for primary. Okay, first step. So we can say P equals primary phase, the first phase. Okay, so during prophase, a couple of things happen. This is actually the longest phase of mitosis because there's a lot of stuff that takes place. So during prophase, our DNA is going to condense to form a chromosome. Okay, and on the next slide, we're going to talk about a little bit more about chromosome structure. Okay, so chromosomes is basically the organized appearance of the DNA. So we're no longer in chromatin, which was like that bowl of spaghetti, but now we are condensing down, we're organizing into chromosomes so that when we start sorting the DNA to be divided, that we make sure every new cell has the correct DNA. While these chromosomes are condensing, these centrioles, remember we learned about those in our eukaryotic organelle unit, those are going to begin to migrate to opposite poles of the cell to form the mitotic spindle, okay? And this is what the little fibers that we're going to need in our next couple of phases. The last thing that happens is that our nuclear envelope is going to break down and disappear. So if you look here at the picture that I have drawn here, you can see that our nuclear envelope is breaking down. You can see inside of our nucleus there are condensed chromosomes in the shape of an X. And lastly, you can see these centrioles that are starting to move apart to the opposite sides of the cell so they can be prepared to start separating the chromosomes when it's time. So what is a chromosome? A chromosome is the organized structure of a DNA, and it looks like this. Oops. It looks like, let me get my little pen here, it looks like an X. Okay, so we're going to label some of the different parts of our chromosome, starting with what's holding them together in the middle. So we have, maybe purple's not the best color, let's use green. Okay, so here is our chromosome. Holding our chromosome together in the middle is called the centromere. This is going to be an important part where our spindle fibers are actually going to attach during metaphase and anaphase. So we have two parts of our chromosome, okay? And these are called, each called a chromatid. Okay, these chromatids are identical. So during the S phase of the cell cycle, remember we replicated all of the DNA, and during that process these chromosomes were replicated. That's what was happening. And so now each side of this chromosome is identical to the other side. Okay, that is important because when we start separating these in a little bit, we want to make sure they get pulled apart correctly so that each new cell ends up with the same DNA. So these are identical, and because they're identical, we not only call them chromatids, we call them together sister chromatids. Because they are identical, okay? So we call them sisters. So sister chromatids. So make sure you have this chromosome drawn and labeled just like I have on the screen. You will need to make sure you know the structure of a chromosome. Alright, so after prophase, we move into metaphase. And for metaphase, I want you to think of the M the M meaning middle. Okay, because in metaphase, this is when our chromosomes line up down the middle of the cell. So you can look down here at the pictures, the picture I have at the bottom. You can see this cell, our nuclear membrane is completely gone. There's no nucleus visible. And you can see our chromosomes lined up down here in the middle of the cell. Now, attached to these chromosomes in the very middle at the centromere are these spindle fibers that are attaching to our chromosomes at the centromere. Okay, so they're not moving yet, they're just lining up down the middle and getting ready to be separated. So step 2, metaphase, the M stands for middle. All right, step 3 is called anaphase. And for anaphase, I want you to think that the A in anaphase stands for apart or away. Okay, so in anaphase, we are pulling our chromosomes apart. So the chromatids are pulled by their centromere. Okay, they're still attached to these spindle fibers. They're being pulled to opposite sides. So if you look down here at the picture, you can see this happening. Now we no longer have the X's in the middle. They're being pulled to opposite directions, to opposite sides of the cell. Okay, so A stands for apart. The final step of mitosis is called telophase. And telophase, I want you to think of the T. The T in telophase stands for 2. Okay, because at the end of telophase, we're going to see 2 new nucleoli appear. So we're going to see we're each going to see 2 nucleuses, but the proper way to say that is nucleoli. So our chromatids are arriving at the opposite sides. We see new membranes, nuclear membranes are forming, and it's going to start uncoiling, going back into the chromatin form, and we can see our 2 nucleoli. Telophase is often called the butt end of mitosis because, unfortunately, telophase kind of looks like a butt when you look at the actual picture of it. So remember, telophase, T is for 2. The butt end of mitosis, 2 nucleoli are appearing, and the last step that we need to do is to complete cell division. So the 4 steps of mitosis are prophase, metaphase, anaphase, and telophase. All you have to remember is PMAT. Okay, I will expect you to know the order of mitosis. So remember PMAT: prophase, metaphase, anaphase, and telophase. Remember, the P stand for primary, the first step. Metaphase stood for middle because our chromosomes were lined up down the middle of our cell. Anaphase stood for apart because our chromosomes, our sister chromatids, were pulled apart to opposite sides. And the T in telophase stood for 2 because now we had 2 new nuclei that were visible within the cell. So that is the end of mitosis, okay? But the cell is not completely divided. So there's one step before we get 2 separate cells. And this step is called cytokinesis. Okay, this happens after the conclusion of mitosis. And this is simply the division of the cytoplasm. Okay, the division of the cytoplasm. So mitosis is the division of the nucleus. Cytokinesis is the division of the cytoplasm. It's going to look a little bit differently in animals versus plants. So in animals, we form what we call a cleavage furrow. So let's look at our picture down here. I have put both the animal cell and plant cell on here so that you can see how these compare. So here is telophase. And you can see at the end of telophase we have this cleavage furrow. This is kind of like there's a contractile ring that's in the center of the cell that starts squeezing the cell basically in half so that we can divide it into 2. So the cleavage furrow constricts and constricts and constricts towards the center until it pinches off into 2 separate cells. Okay, so after cytokinesis in animals, we have 2 separate cells that are no longer attached. Now this is different in plants because in plants instead of separating into two free cells, in cytokinesis we simply form a cell plate right down the middle of the two cells. So we just build basically an extra cell wall in between those two nuclei, which turns into the cell wall separating those two plant cells. Okay, so it's different in plants versus animals. Now, it's important that you understand that cell growth is controlled in our bodies. And so this is done by hormones and things that get triggered for us to make new cells. The purpose of mitosis are two things. Okay, so mitosis, cell growth, cell division, has two things. First, to allow us to grow and get bigger. Okay, obviously you are not the same size as you were when you were born. You have grown, your bone cells have duplicated and gone through this process of mitosis so that you can grow taller, right? Maybe you've built muscle, so your muscle cells go through this process to build more muscle. And the second thing that mitosis and cell division allow is it allows us to repair damage. Okay, so you've probably had a cut, or you've fallen down and scraped your knee or something. You've had some kind of wound. Okay, well, you've noticed probably that that wound healed. Well, that wound healed because of mitosis. So the cells that were wounded, that triggered a hormone to be released, which caused the cells surrounding the wound to begin to go through mitosis and divide to make new cells to start healing up the injury. Okay, so mitosis is what allows us to repair damage and to grow. Okay, so this is a controlled process, right? We control how this works and how often the cells divide and how often and when the cells to stop dividing, etc. So sometimes, though, this can be uncontrolled, and this is not a good thing. So if it's uncontrolled, then the cells are going to divide continuously. So basically, these are not paying attention to the signals that the body is giving. Okay, so they are not listening, they're not responding, and they're just dividing and dividing and dividing over and over and over again. Okay, and what this turns into is cancer. Okay, so cancer is uncontrolled cell growth. Okay, cancer is uncontrolled cell growth, and this is not good, because these cancer cells, all they do is they starve the healthy cells around them. They starve them of nutrients and oxygen, and which makes it difficult for your normal cells to do their job. Okay, so when cancer cells form a mass, we call this a tumor. You've heard of tumors. Tumors are cancer. And malignancy, we say cancer is malignant whenever a cancer cell is able to break free and then forms a new mass somewhere else. That's how cancer is able to spread. Okay, cancer is often hard to detect because it is simply your body cells that have decided not to follow the instructions of the body anymore. This can be from a mutation in the DNA that could be caused by smoking or drug use or anything like that. And so mutations can also be caused by UV radiation, right? That's why tanning beds can be dangerous, because those UV rays literally cause mutations within your skin cells into DNA, which can cause those skin cells to no longer follow the instructions of the body, which can lead to skin cancer called melanoma. So we want to make sure, remember we talked about in the cell cycle, about how cells go through all of these different checkpoints. Remember, those checkpoints are to make sure the cell is healthy, there's no mutations in the DNA, and everything is good before we go through this process. So it's when those cells go through those checkpoints and they aren't caught, okay, that can lead to cancer. All right, so this is the conclusion of our mitosis video. Make sure you have all of your notes recorded, and make sure you go back and study and refresh your memory over all of this, because we will be using this information throughout the upcoming week.